I want to begin by thanking the organizers for inviting me. I have learned a great deal in the last day, and I expect to learn a great deal today and tomorrow. Before I tell you my story today, I would also like to begin with my acknowledgments. I would like to thank and honor my family for their love and support. I would also like to thank my funding sources. So over the last 30 years, and I would also like to acknowledge that I have been funded a long time ago by the pesticide industry. Not anymore. And I would also like to thank all of the students that have been involved in the work over the last 30 years before I begin. About 20 years ago, I was asked by Syngenta, they were Novartis company at the time, to analyze atrazine and whether or not it acted as an endocrine disruptor. I specifically study frogs, and I was asked to determine whether or not this chemical affects frog development. Atrazine is an herbicide, so it's a weed killer. It has been used since 1958, and we use in the United States right now 80 million pounds per year. It's second only to glyphosate as the most commonly used chemical in agriculture. It's used in more than 80 countries, but it's now outlawed in all of Europe. The irony being that the company that makes the chemical is in Switzerland. So they don't use it, but we do in my country. I examined first the African clawed frog, a very common laboratory model for examining developmental biology. And we discovered that atrazine, this chemical, inhibited growth of the voice box, or the larynx. So when the animals were exposed, the males, during development, they could not sing properly, which means they cannot properly reproduce. We also found that the problem was with the gonads, because the larynx of the voice is controlled by the hormone testosterone, the so-called male hormone. Some animals that were exposed to atrazine internally look like this. They might have 2 testes, but then also ovaries, but then also more testes, but then also ovaries, which is not the normal way that frogs develop. Very problematic for the company. We proposed a hypothesis that normally the testis makes testosterone, the hormone that circulates in the male's body, in the male's blood. But we propose that atrazine turns on an enzyme called aromatase. And this enzyme converts testosterone into the female hormone, estrogen. So the males don't develop properly because their testosterone is depleted, but also because they're subsequently feminized because they're making estrogen. So we tested this hypothesis in many ways. We can measure blood levels of testosterone. So here are the control, or the sham, or the placebo-treated animals, males, compared to atrazine-treated males, compared to females. So the testosterone was depleted in these animals. We published this first study in Proceedings in the National Academy of Sciences, maybe 2002. Hermaphroditic demasculinized frogs after exposure to the herbicide atrazine at low ecologically relevant doses. It had a big impact on the company. They didn't like us so much after that. That's okay. We went on to do further studies because we only in the first study examined what happened to the juveniles, so essentially what happened to the larvae, or what happened in their childhood effectively. But we showed that when they become adults, the exposed males, even though they were genetic males, completely turned into females. So for example, there is a gene that's present only in females, and that gene is absent in both of these animals that are reproducing. Both of these animals are genetic male. Even though the one on the bottom, even though is a genetic male, makes eggs and reproduces as a female, even though it was technically born as a genetic male. We then asked some questions with a very simple apparatus. We asked if the males that are exposed to atrazine, can they reproduce properly as a male? And so we really did simple studies in the laboratory by competing atrazine-treated males with controls for females in this reproductive behavior study. And we essentially allow the animals to breed overnight, and then we simply determine, there's markers so that we can tell which ones are which. We simply determine the ability of the atrazine-treated males to compete for the females in this study. And when we conduct this study, we find out that the atrazine-treated males almost never are successful. They almost never get the female. What's more, as we measured the hormone levels in the control or the placebo males compared to the atrazine-treated males, and the testosterone on average is much lower in the atrazine-treated males. What's more important is if you look at the successful males in these experiments, there's a threshold of testosterone that's required for reproduction, and the atrazine-treated males' testosterone is so low, is so suppressed, that they cannot be successful in mating with the female. We did other studies without competition where we exposed just one male and one female per aquarium to test if the males were functional when exposed to atrazine at all. And we measured that function by collecting the eggs after we allowed them some time alone. And we collect the eggs, and then we examine how many are not fertilized, so insufficient sperm, versus ones that are fertilized after a 2-day period. If you conduct these studies, you find out that the control males, the normal unexposed males, have a fertility rate of 85%. So control males fertilize approximately 85% of a female's eggs. Each female lays about 2,000 eggs. Compared to atrazine-treated males, there's only about 15% fertilization. So very, very low fertilization rate if you're exposed to atrazine. We then examine the testis under the microscope to determine why the fertility might be so low. One problem is that the males don't show the behavior, even if there's no competition, even if they're left alone with the female. The other problem is if you examine the testis of a control, an atrazine-treated male, and I will magnify this, in the control, there's sperm inside the testicular tubule. Whereas in the atrazine-treated male, the testis has no sperm, some cellular debris, but not enough testosterone to maintain fertility, to maintain sperm production. Very problematic. We published a second paper also in Proceedings in the National Academy of Sciences. Atrazine induces complete feminization and chemical castration in male African clawed frogs. So more evidence that atrazine has a negative effect on the reproduction. We then examined other species of frogs in the laboratory, and in particular, we examined a North American leopard frog, so an American frog, and we found similar effects. In this case, you can see the testis, so it is a male, but instead of sperm, there are eggs growing inside the testis, and the eggs are bursting through the surface, bursting to outside of the testis, which I would imagine would be very painful. The reason that we examined the North American leopard frog is because we also wanted to determine if these effects occur across species, but also if these effects occur in the wild. So in other words, we were concerned that maybe this was a laboratory artifact. To give you an idea of the level that we're talking about, we produced these effects in the laboratory at 0.1 parts per billion. That's 0.1 micrograms per liter, or 100 nanograms per liter, 100 picograms per mil. Very small amount. That's about 1/1000th of a grain of salt in 2 liters. So not very much. The package of atrazine recommends application at 2.9 to 29 million parts per billion. In other words, a farmer may be using this chemical at levels that are 290 million times higher than we use in my laboratory. You might ask, how much is in the environment? This is a log scale. And from the literature, from global studies from around the world, we find that in agricultural runoff, this is the minimum and maximum detected in agricultural runoff. These are the levels in temporary pools, so temporary water. These are the levels in permanent water, so a pond or a lake or a river. And these are the levels in precipitation, so rain or snow. This is what we use in my laboratory. So all of these habitats would be at risk, several-fold, including rainwater. In the United States, 500,000 pounds of atrazine come down into rainwater every year. It can travel 1,000 kilometers from the point of application. In fact, that is why France banned atrazine, or why the European Union banned atrazine, because when France banned it, it would come over in the rainwater from Germany. So the whole European Union banned it because it travels so far and contaminates so much. The United States Environmental Protection Agency believes that 3 parts per billion is safe in drinking water, 30 times higher than we know to have the biological effect. Because as you just heard, they never really test it for developmental effects. They never really test for effects on young ones, on larvae, or on children. Here's some of our findings, but from the wild, from nature now, not from the laboratory. So these are the kidneys, and these are the testes, which look normal in appearance. But if you examine them from slices under the microscope, and the color's different because of the staining that we use under the microscope. If I blow this up, you'll see that it's not very normal at all. These are eggs instead of sperm inside the testis of an animal from nature that was exposed to atrazine from farming, from agricultural runoff. We conducted a study across the United States. The red shows you where most of the atrazine is used, not very much used here. But we showed that because there's so much contamination, everywhere we found atrazine, we found deformed frogs, reproductive deformities, and vice versa. Now, you just heard about correlation is not causation, but the reason that we got this paper published in Nature is we not only had the correlation, but we did laboratory studies where we took animals from here and raised them in clean water, and you get no reproductive malformity. But if you take animals from this area and raise them in atrazine in a laboratory, you get reproductive malformation. So we had a correlation, but we also had experiments to show cause and effect. And this work was published in Nature. Now I would like to talk more about the broader implications. This is a pond that I photographed in Uganda when I was working there, and it shows what I believe is the arrowroot crop. And I show this because the runoff from that crop is the drinking water for this neighboring village. Now, I don't know that this water is contaminated. My point, though, here is that this photograph shows that environmental health and public health, human health, are one and the same. I think in many developed places, like this is where I live in Oakland, my house is there, we forget the connection between the environment, between nature, and our own health. And we think that somehow we are separate. At least most people in the US do. They don't realize that if the animal health is failing, the environmental health is failing, then there's potentially a problem with humans. Now, I call this from Silent Spring to Silent Night for, I think, a clever reason. Rachel Carson taught in Silent Spring that the death of the birds due to pesticides, mostly DDT, and our silent spring was a warning to humans, I believe that the death of frogs and our silent night, many frog ponds in the U.S. don't sing anymore, it's also a warning. For those of you who are not an expert on frogs and toads, something like 80% of all frogs and toads now are threatened with being endangered. Not just because of pesticides, because of habitat loss, but if the only water you have available is water contaminated with agricultural runoff, which causes these problems, then you expect it to be very much involved in the global amphibian decline. And that is a big concern of mine. A colleague of mine wrote, in ecoepidemiology, the occurrence of an association in more than one species and species population is very strong evidence for causation. My studies are not just correlation, they are controlled laboratory experiments in addition, and we have examined phylogenetically more than one population, more than one species, more than one genera, more than one family of frogs. So I believe we already have very strong evidence. It's not just an artifact of the laboratory, and it's not just an effect on one species. There's broader evidence, though, because other studies from other laboratories around the world have shown similar effects in fish, reptiles, and birds, and mammals. Every vertebrate class that's been examined. I can't study all of these animals, so I did a collaboration with 22 people from around the world representing 12 different countries who all studied atrazine in all of these different animals. And doing that, we find some very interesting results. So here are testes from frogs from my laboratory. Sperm in the testis with atrazine, no sperm. Here's a study from Belgium in fish. Sperm in the testis with atrazine, no sperm. And these are all peer-reviewed. Here's a study from Argentina in reptiles. This is an alligator. Sperm in the testis exposed to atrazine, no sperm. This is work that was done both in Nigeria and in Croatia. I didn't know any of these scientists before this study. Sperm in the testis of a rat with atrazine, no sperm. And this work was done in Pakistan. Sperm in the testis in a bird, quail. When you give it atrazine, no sperm. So very clear assault, very clear damages on the male across animals in studies done in laboratories around the world that we all published together. So do you lose the sperm because atrazine decreases testosterone? That's my hypothesis. This is a study in England, in salmon, in fish. Controls, you give it atrazine, testosterone declines. These are my studies in frogs, testosterone declines. And these are studies in rats, which are used as a model for humans because they're mammals, testosterone declines. So not just my laboratory, not just frogs, not just one study, dozens and dozens and dozens of studies. In humans, we can't do the experiment, of course, not on purpose. But my colleague, Shauna Swan, shows the following. And this is peer-reviewed and published. She examined sperm quality in men in Columbia, Missouri, where they use lots of atrazine, where we grow lots of corn. And she showed that humans that have 0.1 parts per billion atrazine in their urine have very low sperm count and very low fertility. Now, as you just heard, it's just a correlation. But imagine that if you have enough atrazine in your urine, in your body, that chemically castrates a frog or a fish, you as a human also have a low sperm count. It's just a coincidence, but an interesting one. Now, I'm going to change the axes, but I'll leave the data up. And I'm changing the axes because these are atrazine levels in men in California who work in the fields. And I'm going to change the axes again because these are atrazine levels in men in California who apply this chemical. They have levels in their bodies of 2,400 parts per billion. Men who apply atrazine have 24,000 times the atrazine in their urine then we know is associated with low fertility in men. The men who work in the fields in California have 24,000 times the atrazine than we use in my laboratory to produce those effects I showed you. I like to describe it this way. One of these guys could urinate in a bucket. I could use the atrazine in their urine. I could dilute it 24,000 times, and I could use the atrazine in their urine to chemically castrate 24,000 containers of 30 tadpoles each. That's what these men carry in their bodies. This becomes what we call a case of environmental racism, environmental justice, because most of these men are Latinx. They're mostly from Mexico and other Central American countries. 90% of the people who grow our food that we are exposing to chemicals that we know to have these dangers. The other side of the equation, does atrazine turn on aromatase, the enzyme that produces estrogen? And in frogs, it causes egg production. Just fancy words for egg production, egg and egg yolk production. We don't just see that in our frogs. So here's an amphibian with eggs in its testis. But other laboratories show in fish, eggs grow in the testis when atrazine is around, and also in turtles, eggs grow in the testis when atrazine is around. So it's not just frogs, it's not just my laboratory, it's not just my studies. It's more broad. Now mammals, humans, won't grow eggs in their testes. But aromatase is very important in breast cancer and prostate cancer. Behind heart disease, cancer is the #1 cause of death in the US, and the 2 top cancers now, exceeding lung cancer, are breast cancer and prostate cancer. With regards to prostate cancer, they showed in their factory that there's an 8.4-fold higher incidence of prostate cancer in the men who work in their factory packaging atrazine. 8.4 times higher prostate cancer rate. With breast cancer, one study in the US shows, you heard about p-values, with a very strong p-value, 0.0001, that if your well water, if your drinking water is contaminated with atrazine, you are more likely to develop breast cancer. That's just a correlation. But for humans, whole humans in vivo, that's all we'll have. But if you examine rats, and the company did this study, not me, if you examine rats, you find that there's a decrease in testosterone with atrazine, as we've shown in frogs, as others have shown in fish, reptiles, birds. And there's also an increase in blood levels of estrogen in those rats, as we showed in frogs, as others have shown in fish and in reptiles. What's more, the company, they're now called Syngenta, also showed that if you look at the incidence of breast cancer in rats, the incidence of mammary tumors, if you expose them to atrazine, there's a significant increase in mammary tumors and breast cancer in rats. So even though it's only a correlation, in humans, experimentally, we can induce this same disease. In humans also, in my laboratory, and in other laboratories as well, we can take the human cells, we can't do the experiment on the humans, but we can take the human cells and expose them to atrazine. If you do that, if you take a cell from the adrenal gland that doesn't normally make aromatase, doesn't normally produce estrogen, when you expose that cell to atrazine, it starts expressing aromatase. It starts making estrogen, just like we've shown in fish, just like we've shown in amphibians, just like we've shown in reptiles, just like we've shown in birds, just like we've shown in rats. The human biology responds the same way as all these other laboratory animals when you look at the cellular level. So the breast cancer and the prostate cancer are just correlation, but the underlying mechanism is present in all animals, and we can show that. There's the company. I went to visit them. I think they should spell their name with a Y instead of an I, but they don't listen to me. Their company is in a community that's very, very poor. That's 80% black people, 80% African American. They have a pipe that flows into the Mississippi River and then into the Gulf of Mexico. 1.2 million pounds of this chemical flow into the Gulf of Mexico every year in the United States. In a community that mostly looks like this, especially after Hurricane Katrina damaged that community. The reason I bring this up is the following. One of the epidemics that we have in the United States, in my country, is that poor people and black people and Latino people, Hispanic people, are more likely to get many, many diseases relative to Caucasian or white Americans. So, for example, these are the top 13 cancers that you're likely to get in the United States. In red now, 11 of the 13, you're more likely to get statistically if you're black. And that's controlling for income level and access to health care. If you look now at mortality relative to white Americans, to Caucasians, you're more likely to die from 13 out of 13 cancers if you're black. Now, you just heard on stage that cancer is an environmental disease. I was one of the scientists that was part of the presidential panel on cancer. 80% is environmental. So that doesn't mean that there's genetic inferiority in the minority population or the low-income population. It means that if you're an immigrant in the United States, if you're poor, if you're black, if you're Hispanic, you're more likely to live in and more likely to work in areas that we know are associated with chemicals that we know have negative impacts on your health. Even though I study frogs, I've taken up part of the fight because, one, all of the cancer studies we do, none of those cell lines come from black people or poor people or Hispanic people. So even if we find the cure, it's likely irrelevant to the people who are more likely to contract and die from these same diseases. These are the same people that are working in our fields and growing our food and working in our factories. What's more is I have a student who showed that if you take a breast cancer cell and give it atrazine, it starts expressing the gene for aromatase and making estrogen. That's very problematic because breast cancer is driven by estrogen. All of breast cancer starts out estrogen dependent. So once your cell gets damaged, it's estrogen that makes it grow into a tumor. We now know that the local expression of aromatase in that breast cancer, it makes its own estrogen, and that's what causes those damaged cancer cells to turn into tumors and grow. In fact, the number one treatment right now for breast cancer is a chemical called letrozole. It's a drug that works by blocking aromatase and decreasing estrogen so that your cancer stops and can't grow. It no longer has the estrogen to feed it. How much sense does that make when the number one contaminant of drinking water in the US does exactly the opposite? It increases aromatase, increases estrogen, and promotes breast cancer in rats. And the same company up in the 2000s made both chemicals. In the year 2000, and this is the paper I sent to the Obama presidential. I call it the one-stop shop, chemical causes and cures for breast cancer. Because the same company that gave us 80 million pounds of atrazine per year makes the antidote. The same company benefits from letrozole, makes the chemical that causes the breast cancer. And in the year 2000, they discovered this, and they split up into two different companies. Surprise. So I think what's happened is my interest in this aquatic organism has taught me a lot about this aquatic organism. Because we start out in an amniotic fluid. We start out in water, just like my tadpoles. We as humans depend on the same hormones, exactly, testosterone, estrogen, thyroid hormone, cortisol. We depend on the exact same hormones as my frogs do. We now know that the average human is exposed to over 300 synthetic chemicals before they leave the womb. The best thing about atrazine is we know what it does. For the other 80,000 chemicals that we've invented, we have no idea what they do in this regard, as you just heard Dr. Liu talk about. I want to show you now a series of studies that weren't mine. I'm one of the few scientists who brags about things that I didn't do. These are other people's work. And I'll tell you why they impacted me. These are studies on rats. And first, let me tell you, I study frogs because I love frogs. I've loved frogs since I was a little boy. Nobody studies rats because they love rats. There's nobody walking around saying, I'm a little boy who loves rats. We study rats because we want to learn about humans, period. Studies have shown that atrazine causes prostate and mammary cancer in rats. They're published, peer-reviewed. Other studies have shown that atrazine causes immune failure in rats. We showed the same thing in my laboratory in frogs. Other studies have shown that when rats are exposed in utero, they suffer neural damage, just like you heard for glyphosate. Other studies have shown the following, and here's the study that affected me the most. If you expose pregnant rats to atrazine, they're more likely to have an abortion because of the hormone imbalance. In fact, an EPA laboratory published that study. A second EPA laboratory showed that if those rats that don't abort, the sons are born with prostate disease. Atrazine causes the sons to be born with the prostate of an old man. A third EPA laboratory showed that if you exposed the rats of those that don't abort, the daughters are born with mammary development, poor breast development. And when those rats grow up, their offspring have retarded growth and development because the mother can't make enough milk. That rat on the bottom never saw atrazine. The rat on the bottom was affected by atrazine that its grandmother was exposed to. And you just heard about this, how we're affecting the future. That rat on the bottom never saw atrazine. The rat on the bottom was affected by atrazine that its grandmother was exposed to. The rat on the bottom never saw atrazine. The rat on the bottom was affected by atrazine that its grandmother was exposed to. 2 generations down. And so when I think, this is my wife and my daughter, when I think about my kids, when I think about the fact that my grandchildren could be affected by chemicals that we're using today, chemicals that we're putting on our food, your grandchildren could be affected by chemicals that we're using today. It makes me realize that we have a big responsibility. And we have an expression in the English called preaching to the choir. I'm sure everybody here already knows this. And I'm going to apologize. I have some images that I will warn you. If you don't want to see, you can cover your eyes. But here's studies already. Again, it's just correlation. But with all the experimental evidence we have, we already know that if you get pregnant during peak atrazine contamination, you're more likely to have babies with birth defects. It's just a correlation. Some of those birth defects include, and these are published papers, that one that concluded maternal exposure to surface water atrazine is associated with fetal gastroenteritis. I didn't know what that is. And it's a graphic image. You can cover your eyes before I show it. Gastroenteritis is when the baby's born with the intestines outside of the body. And there are areas in Hawaii and London where there's lots of atrazine, where this rare condition shows up, and as many as 7 births on one street. Other studies show that atrazine causes, and these aren't my studies, causes choanal atresia. And again, you can cover your eyes if you don't want to see. Choanal atresia is where the baby's born with a hole in his face. The nasal and oral cavity don't close properly. Other studies, and again, I apologize if this is culturally insensitive or you don't want to see, but other studies show that if you are pregnant with a boy and you're exposed to atrazine, this is a case-controlled study of maternal residential atrazine exposure and male genital malformations. And again, close your eyes if I'm being culturally insensitive or if you don't want to see the image. If you have a boy and you're exposed to atrazine, you're more likely to have a baby with hypospadias. That's where the urethra doesn't end through the penis. You're more likely to have a baby with cryptorchidism. That's where the testicles don't descend into the scrotum, you're more likely to have a baby with micropenis. That's where the penis doesn't grow. And what interests me is that male genitals depend on testosterone. This is just a correlation. Male genitals depend on testosterone. And if you're exposed to a chemical that lowers testosterone, you have a baby that looks like it didn't have enough testosterone. And we know that estrogens can cause these malformations. And if you're exposed to atrazine, you have a baby that looks like it was exposed to too much estrogen. So it's just correlation, but there's way too much evidence to support causation. Way too much evidence for me to not speak out against the company that paid me to discover these things. Now, I also work with the Environmental Protection Agency. Here's what they said about my work 2 years ago. They wrote, quote, "A monetary value is assigned to disease, impairments, and shortened lives, and weighed against the benefits of keeping a chemical in use." So they're not saying that there's no problem with atrazine. They're not saying, they were refuting my findings. They're not refuting my findings. 20 years later, they're saying, sorry if I speak fast, but I get excited when I talk about this. They're saying, we know there's a problem, but we put a price, and that price is on minorities and poor people, because the people who are exposed are the people who are exposed the most, we all are the people in the factories and the people working in the fields. The EPA says that's the price that they pay for somebody else to get rich. I say somebody else to get rich because of the following. This is my state, California. I live right about there, if you care. California is the 5th or 7th, depending on who you talk to, richest country in the world. If we were our own country, we'd be the 5th richest country in the world. You know why? Not because of Hollywood, not because of technology, because of agriculture. 1 in 10 jobs are in agriculture. 30% of the land is in agriculture. 350 agricultural products. And here's one that I didn't know. 50%. Half of the United States' food comes from California. Half of the whole country's food comes from not just California, a very small part of California. We use more pesticides than any other state, and 90% of the workers that feed half of the country are Hispanic, mostly Mexican. Now, in red now, these are the top 10 counties for agriculture in California. These are the counties that make us the 5th richest country in the world. Who's paying that price? Do you know where the 30 poorest towns are in California? So the people who make us the 5th largest economy in the world are the targets of chemicals that we know adversely affects their health care, that leads to life expectancy. You know what the life expectancy of a migrant farm worker is? Between 40 and 50. And the 5th largest economy in the world. Our problems also, this is a national park from my hometown in South Carolina. Turns out it's contaminated with atrazine. It's a swamp. You know how many frog and reptile species are supposed to be protected in that park? It's also a swamp. That means that the surrounding human community is all black, poor people, because nobody wants to live in a swamp. Here are the other problems, and you just heard this from Dr. Heron. It is not about feeding the world. It is not about growing food. This is a graph of where all of the corn goes in the United States. What's missing from this graph? Food. Food. We eat so little of the corn that it doesn't even appear in the graph. We eat less than 2% of the corn. So we're using this chemical to increase corn yield by 1.2%. That's kilograms per acre you just heard about. We're using this chemical to increase corn yield by 1.2%, of which we eat less than 2%, while 20% of the world dies because it doesn't have access to staples. This has nothing to do with feeding the world. If you look at the United States, again, here's a map. The green shows states where the majority of the agricultural income comes from feeding people. The red, remember this is where all the atrazine was used, are states where the majority of the agricultural income goes to feeding animals. or other sources. Most of these are around 100%. So all of the atrazine that we're using has nothing to do with feeding the world. Has nothing to do with feeding the United States, in fact. It has to do with making money. Here's a large part of the problem. Who's ever seen this figure? And blue now are seed companies. And red now, and now there's only four big ones, are the chemical companies that own 90% of the seeds that we use globally to grow our food. That means that the goal is not to produce seed, is not to produce food. The goal is to sell chemicals. And when I was a freshman in college, a few years ago, the promise of GMO was more nutritious grains and frost-resistant strawberries, drought-resistant wheat. 90% of GMO now is to make plants that require more chemical. And this is a large part of the problem. The companies that own our seed, their goal is to sell chemical, not to grow food. Another large part of the problem is Obama. Every agency in the United States, I believe, that regulates food is run by somebody from the chemical industry. And this was under the good president. I can't imagine where we're headed now. But it doesn't make me optimistic. Thank you again for the invitation. And thank you for listening and your time.